W 2. Self. A tear is the most abundant substance in living systems, making up 70% or more of the weight of most organisms. The first living organisms on Earth doubtless arose in an aqueous environment, and the course of evolution has been shaped by the properties of the aqueous medium in which life began. This chapter begins with descriptions of the physical and chemical properties of water, to which all aspects of cell structure and function are adapted. The attractive forces between water molecules and the slight tendency of water to ionize are of crucial importance to the structure and function of biomolecules. We review the topic of ionization in terms of equilibrium constants, pH, and titration curves, and consider how aqueous solutions of weak acids, or bases, and their salts act as buffers against pH changes in biological systems. The water molecule and its ionization products, H+ and OH, profoundly influence the structure, self-assembly, and properties of all cellular components, including proteins, nucleic acids, and lipids. The non-covalent interactions responsible for the strength and specificity of recognition among biomolecules are decisively influenced by water's properties as a solvent, including its ability to form hydrogen bonds with itself and with solutes. 2.1 Weak interactions in aqueous systems. Hydrogen bonds between water molecules provide the cohesive forces that make water a liquid at room temperature and a crystalline solid, ice, with a highly ordered arrangement of molecules at cold temperatures. Polar biomolecules dissolve readily in water, because they can replace water-water interactions with more energetically favorable water-solute interactions. In contrast, nonpolar biomolecules are poorly soluble in water, because they interfere with water-water interactions, but are unable to form water-solute interactions. In aqueous solutions, nonpolar molecules tend to cluster together. Hydrogen bonds and ionic, hydrophobic, Greek, water-fearing, and van der Waals interactions are individually weak, but collectively they have a very significant influence on the three-dimensional structures of proteins, nucleic acids, polysaccharides, and membrane lipids. Hydrogen bonding gives water its unusual properties. Water has a higher melting point, boiling point, and heat of vaporization than most other common solvents, Table 2:1. These unusual properties are a consequence of attractions between adjacent water molecules that give liquid water great internal cohesion. A look at the electron structure of the H2O molecule reveals the cause of these intermolecular attractions. Each hydrogen atom of a water molecule shares an electron pair with the central oxygen atom. The geometry of the molecule is dictated by the shapes of the outer electron orbitals of the oxygen atom, which are similar to the sp3 bonding orbitals of carbon. See Fig. 1-16. These orbitals describe a rough tetrahedron, with a hydrogen atom at each of two corners and unshared electron pairs at the other two corners. Fig. 2-1a. The HOH bond angle is 104.5 degrees, slightly less than the 109.5 degrees of a perfect tetrahedron because of crowding by the non-bonding orbitals of the oxygen atom. Table 2. Heat of common melting, heat of water, propane, butane, acid, benzene, chloroform. Of the heat, the oxygen nucleus attracts electrons more strongly than does the hydrogen nucleus, a proton, that is, oxygen is more electronegative. This means that the shared electrons are more often in the vicinity of the oxygen atom than of the hydrogen. The result of this unequal electron sharing is two electric dipoles in the water molecule, one along each of the HO bonds. Each hydrogen atom bears a partial positive charge, delta plus, and the oxygen atom bears a partial negative charge equal in magnitude to the sum of the two partial positives, 2 delta. As a result, there is an electrostatic attraction between the oxygen atom of one water molecule and the hydrogen of another, Fig. 2. 1b, called a hydrogen bond. Throughout this book, we represent hydrogen bonds with three parallel blue lines, as in Figure 2-1b. Figure 2. Hydrogen bonds are relatively weak. Those in liquid water have a bond dissociation energy, the energy required to break a bond, of about 23 kJ/mol, compared with 470 kJ/mol for the covalent OH bond in water, or 348 kJ/mol for a covalent CC bond. The hydrogen bond is about 10% covalent, due to overlaps in the bonding orbitals, and about 90% electrostatic. At room temperature, the thermal energy of an aqueous solution, the kinetic energy of motion of the individual atoms and molecules, is of the same order of magnitude as that required to break hydrogen bonds. When water is heated, the increase in temperature reflects the faster motion of individual water molecules. At any given time, most of the molecules in liquid water are hydrogen bonded, but the lifetime of each hydrogen bond is just 1 to 20 picoseconds, 1 ps equals 1012 s. When one hydrogen bond breaks, another hydrogen bond forms, with the same partner or a new one, within 0.1 ps. The apt phrase flickering clusters has been applied to the short-lived groups of water molecules interlinked by hydrogen bonds in liquid water. The sum of all the hydrogen bonds between H2O molecules confers great internal cohesion on liquid water. Extended networks of hydrogen-bonded water molecules also form bridges between solutes, proteins and nucleic acids, for example, that allow the larger molecules to interact with each other over distances of several nanometers without physically touching. The nearly tetrahedral arrangement of the orbitals about the oxygen atom, Fig. 2-1a, allows each water molecule to form hydrogen bonds with as many as 4 neighboring water molecules. In liquid water at room temperature and atmospheric pressure, however, water molecules are disorganized and in continuous motion, so that each molecule forms hydrogen bonds with an average of only 3.4 other molecules. In ice, on the other hand, each water molecule is fixed in space and forms hydrogen bonds with a full complement of 4 other water molecules to yield a regular lattice structure, Fig. 2-2. Hydrogen bonds account for the relatively high melting point of water, because much thermal energy is required to break a sufficient proportion of hydrogen bonds to destabilize the crystal lattice of ice, Table 2-1. When ice melts or water evaporates, heat is taken up by the system. Figure 2-2 hydrogen. During melting or evaporation, the entropy of the aqueous system increases as the highly ordered arrays of water molecules in ice relax into the less orderly hydrogen-bonded arrays in liquid water, or into the wholly disordered gaseous state. At room temperature, both the melting of ice and the evaporation of water occur spontaneously. The tendency of the water molecules to associate through hydrogen bonds is outweighed by the energetic push toward randomness. Recall that the free energy change, delta G, must have a negative value for a process to occur spontaneously. Delta G equals delta HT delta S, where delta G represents the driving force, delta H the enthalpy change from making and breaking bonds, and delta S the change in randomness. Because delta H is positive for melting and evaporation, it is clearly the increase in entropy, delta S, that makes delta G negative and drives these changes. Water forms hydrogen bonds with polar solutes. Hydrogen bonds are not unique to water. They readily form between an electronegative atom, the hydrogen acceptor, usually oxygen or nitrogen, and a hydrogen atom covalently bonded to another electronegative atom, the hydrogen donor, in the same or another molecule Fig. 2-3. Hydrogen atoms covalently bonded to carbon atoms do not participate in hydrogen bonding, because carbon is only slightly more electronegative than hydrogen, and thus the CH bond is only very weakly polar. The distinction explains why butane, CH3, CH2, 2CH3, has a boiling point of only 0.5 degrees C, whereas butanol, CH3, CH2, 2CH2OH, has a relatively high boiling point of 117 degrees Celsius. Butanol has a polar hydroxyl group and thus can form intermolecular hydrogen bonds. Uncharged but polar biomolecules such as sugars dissolve readily in water, because of the stabilizing effect of hydrogen bonds between the hydroxyl groups or carbonyl oxygen of the sugar and the polar water molecules. Alcohols, aldehydes, ketones, and compounds containing NH bonds all form hydrogen bonds with water molecules, Fig. 2-4, and tend to be soluble in water. Figure 2-3. Figure 2-4. Figure 2-5. Hydrogen bonds are strongest when the bonded molecules are oriented to maximize electrostatic interaction, which occurs when the hydrogen atom and the two atoms that share it are in a straight line, that is, when the acceptor atom is in line with the covalent bond between the donor atom and H. Fig. 2-5. This arrangement puts the positive charge of the hydrogen ion directly between the two partial negative charges. Hydrogen bonds are thus highly directional and capable of holding two hydrogen-bonded molecules or groups in a specific geometric arrangement. As we shall see, this property of hydrogen bonds confers very precise three-dimensional structures on protein and nucleic acid molecules, which have many intramolecular hydrogen bonds. Water interacts electrostatically with charged solutes. Water is a polar solvent. It readily dissolves most biomolecules, which are generally charged or polar compounds. Table 2-2. Compounds that dissolve easily in water are hydrophilic, Greek, water-loving. In contrast, nonpolar solvents such as chloroform and benzene are poor solvents for polar biomolecules, but easily dissolve those that are hydrophobic nonpolar molecules such as lipids and waxes. Water dissolves salts such as NaCl by hydrating and stabilizing the Na+ and Cl ions, weakening the electrostatic interactions between them, and thus counteracting their tendency to associate in a crystalline lattice, Fig. 2-6. Water also readily dissolves charged biomolecules, including compounds with functional groups, such as ionized carboxylic acids, COO, protonated amines, and phosphate esters or anhydrides. Water replaces the solute-solute hydrogen bonds linking these biomolecules to each other with solute water-hydrogen bonds, thus screening the electrostatic interactions between solute molecules. Water is effective in screening the electrostatic interactions between dissolved ions because it has a high dielectric constant, a physical property that reflects the number of dipoles in a solvent. The strength or force F of ionic interactions in a solution depends on the magnitude of the charges, Q, the distance between the charged groups, R, and the dielectric constant, epsilon, which is dimensionless, of the solvent in which the interactions occur. For water at 25 degrees Celsius, epsilon is 78.5, and for the very nonpolar solvent benzene, epsilon is 4.6. Thus, ionic interactions between dissolved ions are much stronger in less polar environments. The dependence on R2 is such that ionic attractions or repulsions operate only over short distances in the range of 10 to 40 nanometers, depending on the electrolyte concentration, when the solvent is water. Table 2. Amphipathic forms. Figure 2-6. Entropy increases as crystalline substances dissolve. As a salt such as NaCl dissolves, the Na+ and Cl ions leaving the crystal lattice acquire far greater freedom of motion, Fig. 2-6. The resulting increase in entropy, randomness, of the system is largely responsible for the ease of dissolving salts such as NaCl in water. In thermodynamic terms, formation of the solution occurs with a favorable free energy change, delta G equals delta HT delta S, where delta H has a small positive value and T delta S a large positive value. Thus delta G is negative. Nonpolar gases are poorly soluble in water. The molecules of the biologically important gases CO2, O2, and N2 are nonpolar. In O2 and N2, electrons are shared equally by both atoms. In CO2, each C equals O bond is polar, but the two dipoles are oppositely directed and cancel each other, Table 2-3. The movement of molecules from the disordered gas phase into aqueous solution constrains their motion, and the motion of water molecules, and therefore represents a decrease in entropy. The nonpolar nature of these gases and the decrease in entropy when they enter solution combine to make them very poorly soluble in water, Table 2-3. Some organisms have water-soluble carrier proteins, hemoglobin and myoglobin, for example, that facilitate the transport of O2. Carbon dioxide forms carbonic acid, H2CO3, in aqueous solution, and is transported as the bicarbonate ion. Either free bicarbonate is very soluble in water, 100 G/L at 25 degrees Celsius, or bound to hemoglobin. Three other gases, NH3, NO, and H2S, also have biological roles in some organisms. These gases are polar, dissolve readily in water, and ionize in aqueous solution. Table 2. Gas structure. Nitrogen N, oxygen O, ammonia, hydrogen A. The error. Non-polar compounds force energetically unfavorable changes in the structure of water. When water is mixed with benzene or hexane, two phases form, neither liquid is soluble in the other. Non-polar compounds such as benzene and hexane are hydrophobic. They are unable to undergo energetically favorable interactions with water molecules, and they interfere with the hydrogen bonding among water molecules. All molecules or ions in aqueous solution interfere with the hydrogen bonding of some water molecules in their immediate vicinity, but polar or charged solutes, such as NaCl, compensate for lost water-water hydrogen bonds by forming new solute-water interactions. The net change in enthalpy, ΔH, for dissolving these solutes is generally small. Hydrophobic solutes, however, offer no such compensation, and their addition to water may therefore result in a small gain of enthalpy. The breaking of hydrogen bonds between water molecules takes up energy from the system, requiring the input of energy from the surroundings. In addition to requiring this input of energy, dissolving hydrophobic compounds in water produces a measurable decrease in entropy. Water molecules in the immediate vicinity of a nonpolar solute are constrained in their possible orientations as they form a highly ordered cage-like shell around each solute molecule. These water molecules are not as highly oriented as those in clathrates, crystalline compounds of nonpolar solutes in water, but the effect is the same in both cases. The ordering of water molecules reduces entropy. The number of ordered water molecules, and therefore the magnitude of the entropy decrease, is proportional to the surface area of the hydrophobic solute enclosed within the cage of water molecules. The free energy change for dissolving a nonpolar solute in water is thus unfavorable. ΔG equals ΔHT ΔS, where ΔH has a positive value, ΔS has a negative value, and ΔG is positive. Figure 2. Amphipathic compounds contain regions that are polar or charged, and regions that are nonpolar, Table 2:2. When an amphipathic compound is mixed with water, the polar, hydrophilic region interacts favorably with the water and tends to dissolve, but the nonpolar, hydrophobic region tends to avoid contact with the water, Fig. 2-7a. The nonpolar regions of the molecules cluster together to present the smallest hydrophobic area to the aqueous solvent, and the polar regions are arranged to maximize their interaction with the solvent, Fig. 2-7b, a phenomenon called the hydrophobic effect. These stable structures of amphipathic compounds in water, called micelles, may contain hundreds or thousands of molecules. The forces that hold the nonpolar regions of the molecules together are sometimes referred to as hydrophobic interactions, although this terminology can be confusing because the strength of the interactions is not due to any intrinsic attraction between nonpolar moieties. Rather, it results from the systems achieving the greatest thermodynamic stability by minimizing the number of ordered water molecules required to surround hydrophobic portions of the solute molecules. Figure 2. Many biomolecules are amphipathic. Proteins, pigments, certain vitamins, and the sterols and phospholipids of membranes all have both polar and nonpolar surface regions. Structures composed of these molecules are stabilized by the hydrophobic effect, which favors aggregation of the nonpolar regions. The hydrophobic effect on interactions among lipids, and between lipids and proteins, is the most important determinant of structure in biological membranes. The aggregation of nonpolar amino acids in protein interiors, driven by the hydrophobic effect, also stabilizes the three-dimensional structures of proteins. Hydrogen bonding between water and polar solutes also causes an ordering of water molecules, but the energetic effect is less significant than with nonpolar solutes. Disruption of ordered water molecules is part of the driving force for binding of a polar substrate, reactant, to the complementary polar surface of an enzyme. Entropy increases as the enzyme displaces ordered water from the substrate and as the substrate displaces ordered water from the enzyme surface. Fig. 2-8. Van der Waals interactions are weak interatomic attractions. When two uncharged atoms are brought very close together, their surrounding electron clouds influence each other. Random variations in the positions of the electrons around one nucleus may create a transient electric dipole, which induces a transient, opposite electric dipole in the nearby atom. The two dipoles weakly attract each other, bringing the two nuclei closer. These weak attractions are called van der Waals interactions, also known as London forces. As the two nuclei draw closer together, their electron clouds begin to repel each other. At the point where the net attraction is maximal, the nuclei are said to be in van der Waals contact. Each atom has a characteristic van der Waals radius, a measure of how close that atom will allow another to approach. Table 2-4. In the space-filling molecular models shown throughout this book, the atoms are depicted in sizes proportional to their van der Waals radii. Table 2. Single bond elements. Element H. Weak interactions are crucial to macromolecular structure and function. I believe that as the methods of structural chemistry are further applied to physiological problems, it will be found that the significance of the hydrogen bond for physiology is greater than that of any other single structural feature. Linus Pauling, The Nature of the Chemical Bond, 1939. The non-covalent interactions we have described, hydrogen bonds and ionic, hydrophobic, and van der Waals interactions, Table 2-5, are much weaker than covalent bonds. An input of about 350 kJ of energy is required to break a mole of 6 × 10^23 cc single bonds, and about 410 kJ to break a mole of CH bonds, but as little as 4 kJ is sufficient to disrupt a mole of typical van der Waals interactions. Interactions driven by the hydrophobic effect are also much weaker than covalent bonds, although they are substantially strengthened by a highly polar solvent, a concentrated salt solution, for example. Ionic interactions and hydrogen bonds are variable in strength, depending on the polarity of the solvent and the alignment of the hydrogen-bonded atoms, but they are always significantly weaker than covalent bonds. In aqueous solvent at 25 degrees Celsius, the available thermal energy can be of the same order of magnitude as the strength of these weak interactions, and the interaction between solute and solvent, water, molecules is nearly as favorable as solute-solute interactions. Consequently, hydrogen bonds and Ionic, hydrophobic, and van der Waals interactions are continually forming and breaking. Table 2 to hydrogen bond between ionic repulsion. Although these four types of interactions are individually weak relative to covalent bonds, the cumulative effect of many such interactions can be very significant. For example, the non-covalent binding of an enzyme to its substrate may involve several hydrogen bonds and one or more ionic interactions, as well as hydrophobic and van der Waals interactions. The formation of each of these weak bonds contributes to a net decrease in the free energy of the system. We can calculate the stability of a non-covalent interaction, such as the hydrogen bonding of a small molecule to its macromolecular partner, from the binding energy, the reduction in the energy of the system when binding occurs. Stability, as measured by the equilibrium constant, see below, of the binding reaction, varies exponentially with binding energy. To dissociate two biomolecules, such as an enzyme and its bound substrate, that are associated non-covalently through multiple weak interactions, all these interactions must be disrupted at the same time. Because the interactions fluctuate randomly, such simultaneous disruptions are very unlikely. Therefore, 5 or 20 weak interactions bestow much greater molecular stability than would be expected intuitively from a simple summation of small binding energies. Macromolecules such as proteins, DNA, and RNA contain so many sites of potential hydrogen bonding or ionic, van der Waals, or hydrophobic interactions that the cumulative effect of the many small binding forces can be enormous. For macromolecules, the most stable, that is, the native, structure is usually that in which weak interactions are maximized. The folding of a single polypeptide or polynucleotide chain into its three-dimensional shape is determined by this principle. The binding of an antigen to a specific antibody depends on the cumulative effects of many weak interactions. As noted earlier, the energy released when an enzyme binds non-covalently to its substrate is the main source of the enzyme's catalytic power. The binding of a hormone or a neurotransmitter to its cellular receptor protein is the result of multiple weak interactions. One consequence of the large size of enzymes and receptors, relative to their substrates or ligands, is that their extensive surfaces provide many opportunities for weak interactions. At the molecular level, the complementarity between interacting biomolecules reflects the complementarity and weak interactions between polar and charged groups, and the proximity of hydrophobic patches on the surfaces of the molecules. When the structure of a protein such as hemoglobin, Fig. 2-9, is determined by X-ray crystallography, see box 4-5, water molecules are often found to be bound so tightly that they are part of the crystal structure. The same is true for water in crystals of RNA or DNA. These bound water molecules, which can also be detected in aqueous solutions by nuclear magnetic resonance, have distinctly different properties from those of the bulk water of the solvent. They are, for example, not osmotically active, see below. For many proteins, tightly bound water molecules are essential to their function. In a key reaction in photosynthesis, for example, protons flow across a biological membrane as light drives the flow of electrons through a series of electron-carrying proteins, see Fig. 20-21. One of these proteins, cytochrome F, has a chain of 5 bound water molecules, Fig. 2 to 10, that may provide a path for protons to move through the membrane by a process known as proton hopping, described below. Another such light-driven proton pump, bacteriorhodopsin, almost certainly uses a chain of precisely oriented bound water molecules in the transmembrane movement of protons, see Fig. 20-29b. Tightly bound water molecules can also form an essential part of a protein's ligand binding site. In a bacterial arabinose binding protein, for example, 5 water molecules form hydrogen bonds that provide critical crosslinks between the sugar, arabinose, and the amino acid residues in the sugar binding site, Fig. 2 to 11. Figure 2 to 9 water binding structure, visible only in the beta. Solutes affect the colligative properties of aqueous solutions. Solutes of all kinds alter certain physical properties of the solvent, water, its vapor pressure, boiling point, melting point, freezing point, and osmotic pressure. These are called colligative properties, colligative meaning tied together, because the effect of solutes on all 4 properties has the same basis. The concentration of water is lower in solutions than in pure water. The effect of solute concentration on the colligative properties of water is independent of the chemical properties of the solute. It depends only on the number of solute particles, molecules or ions, in a given amount of water. For example, a compound such as NaCl, which dissociates in solution, has an effect on osmotic pressure that is twice that of an equal number of moles of a non-dissociating solute such as glucose. Figure 2 to 10 water chain. Water molecules tend to move from a region of higher water concentration to one of lower water concentration, in accordance with the tendency in nature for a system to become disordered. When 2 different aqueous solutions are separated by a semi-permeable membrane, one that allows the passage of water, but not solute molecules, water molecules, diffusing from the region of higher water concentration to the region of lower water concentration produce osmotic pressure, Fig. 2 to 12. Osmotic pressure pi, measured as the force necessary to resist water movement, Fig. 2-12c, is approximated by the Van 't Hoff equation. Pi equals ICRT, in which R is the gas constant and T is the absolute temperature. The symbol I is the Van 't Hoff factor, which is a measure of the extent to which the solute dissociates into 2 or more ionic species. The term IC is the osmolarity of the solution, the product of the Van 't Hoff factor I and the solute's molar concentration C. In dilute NaCl solutions, the solute completely dissociates into Na plus and Cl, doubling the number of solute particles, and thus I equals 2. For all non-ionizing solutes, I equals 1. For solutions of several N solutes, pi is the sum of the contributions of each species. Pi equals RT, I1C1 plus I2C2 plus I3C3 plus ing. Figure 2 to 11. Molecules are essential components of the osmosis, water movement across a semi-permeable membrane driven by differences in osmotic pressure, is an important factor in the life of most cells. Plasma membranes are more permeable to water than to most other small molecules, ions, and macromolecules because protein channels, aquaporins, see Fig. 11-43, in the membrane selectively permit the passage of water. Solutions of osmolarity equal to that of a cell's cytosol are said to be isotonic relative to that cell. Surrounded by an isotonic solution, a cell neither gains nor loses water, Fig. 2-13. In a hypertonic solution, one with higher osmolarity than that of the cytosol, the cell shrinks as water moves out. In a hypotonic solution, one with a lower osmolarity than the cytosol, the cell swells as water enters. In their natural environments, cells generally contain higher concentrations of biomolecules and ions than their surroundings, so osmotic pressure tends to drive water into cells. If not somehow counterbalanced, this inward movement of water would distend the plasma membrane and eventually cause bursting of the cell, osmotic lysis. Figure 2. Water and the semi-permeable membrane allows the passage of water. Figure 2-13. Membrane in the direct. Several mechanisms have evolved to prevent this catastrophe. In bacteria and plants, the plasma membrane is surrounded by a non-expandable cell wall of sufficient rigidity and strength to resist osmotic pressure and prevent osmotic lysis. Certain freshwater protists that live in a highly hypotonic medium have an organelle, contractile vacuole, that pumps water out of the cell. In multicellular animals, blood plasma and interstitial fluid, the extracellular fluid of tissues, are maintained at an osmolarity close to that of the cytosol. The high concentration of albumin and other proteins in blood plasma contributes to its osmolarity. Cells also actively pump out Na+ and other ions into the interstitial fluid to stay in osmotic balance with their surroundings. Because the effect of solutes on osmolarity depends on the number of dissolved particles, not their mass, macromolecules, proteins, nucleic acids. Polysaccharides have far less effect on the osmolarity of a solution than would an equal mass of their monomeric components. For example, a gram of a polysaccharide composed of 1,000 glucose units has the same effect on osmolarity as a milligram of glucose. Storing fuel as polysaccharides, starch or glycogen, rather than as glucose or other simple sugars avoids an enormous increase in osmotic pressure in the storage cell. Plants use osmotic pressure to achieve mechanical rigidity. The very high solute concentration in the plant cell vacuole draws water into the cell, Fig. 2-13, but the non-expandable cell wall prevents swelling. Instead, the pressure exerted against the cell wall, turgor pressure, increases, stiffening the cell, the tissue, and the plant body. When the lettuce in your salad wilts, it is because loss of water has reduced turgor pressure. Osmosis also has consequences for laboratory protocols. Mitochondria, chloroplasts, and lysosomes, for example, are enclosed by semi-permeable membranes. In isolating these organelles from broken cells, biochemists must perform the fractionations in isotonic solutions, see Fig. 1-9, to prevent excessive entry of water into the organelles and the swelling and bursting that would follow. Buffers used in cellular fractionations commonly contain sufficient concentrations of sucrose or some other inert solute to protect the organelles from osmotic lysis. Worked example 2-1. organelle I. Suppose the major solution we want pi equals RT, where R is the gas, the osmotic sucrose. In this sucrose, the osmotic, so the worked organelle. Suppose solution sucrose. Similarly, glycogen for a or summary systems. Summary 2.1 Weak interactions in aqueous systems. The very different electronegativities of H and O make water a highly polar molecule, capable of forming hydrogen bonds with itself and with solutes. Hydrogen bonds are fleeting, primarily electrostatic, and weaker than covalent bonds. Water is a good solvent for polar, hydrophilic solutes, with which it forms hydrogen bonds, and for charged solutes, with which it interacts electrostatically. Nonpolar, hydrophobic compounds dissolve poorly in water. They cannot hydrogen bond with the solvent, and their presence forces an energetically unfavorable ordering of water molecules at their hydrophobic surfaces. To minimize the surface exposed to water, nonpolar and amphipathic compounds such as lipids form aggregates, micelles, in which the hydrophobic moieties are sequestered in the interior, an association driven by the hydrophobic effect, and only the more polar moieties interact with water. Weak, non-covalent interactions, in large numbers, decisively influence the folding of macromolecules such as proteins and nucleic acids. The most stable macromolecular conformations are those in which hydrogen bonding is maximized within the molecule and between the molecule and the solvent, and in which hydrophobic moieties cluster in the interior of the molecule away from the aqueous solvent. The physical properties of aqueous solutions are strongly influenced by the concentrations of solutes. When two aqueous compartments are separated by a semi-permeable membrane, such as the plasma membrane separating a cell from its surroundings, water moves across that membrane to equalize the osmolarity in the two compartments. This tendency for water to move across a semi-permeable membrane produces the osmotic pressure.